Hello everyone and welcome to this second session in our open data theme. This is the keynote speaker today. We'd like to again take a minute to welcome everyone from our global audience, whether you're joining us from the OHBM platform or one of our external streaming platforms. Thank you for being with us again today. To highlight the links at the bottom of the slide here, these are the different ways that you can get in touch with us. There's lots of information on our website, ohbm.github.io/OSR 2020. There's the full program and lots more information about the themes and other talks you can expect during the week. If you're on Twitter, you can reach us at OHBM Open, and you can use the hashtags OHBM 2020 and hashtag OSR. If you're using Mattermost, you can have a general discussion with us on HBM Open Science Room channel. And if you want to have a discussion about this specific theme, we've got the channel OSR Open Data.
So this specific session in open data is called the Keynote. And we are going to have, with the speaker, an in-depth look at handling of a big data set and what that means for us for the future of open data. This is going to be one talk, 25 minutes long, and then we'll have a 15-minute Q&A session with the speaker live, if that speaker is available in this time zone hub. Yes.
So we are using Crowdcast for our main Q&A during the talk. If you're using it on Crowdcast, then please put your questions in the question function, and don't forget to upvote other people's questions if you really want to see that one answered. If you're on the HBM platform, you can use the chat function there during the talk. If you'd like to continue the discussion after the session, you can do that on Mattermost on our channel for this theme. And if you can, tag the speakers so they can get your questions, that would be great. We also would love for you to fill out the feedback poll on Crowdcast before you leave the session, if you can. We're going to be using this data to help us build the best OSR we can, see what you're really into, and also to give individual speakers some feedback about their content. And finally, just to remind you all of the code of conduct, we would love for you to treat each other with kindness and respect in every interaction you have, whether it's online or in one of the social sessions. And again, a reminder that we all agreed to abide by the code of conduct when we registered for the meeting. And if you see or experience any violations of the code of conduct, please do follow the reporting guidelines that are outlined in the document on the HBM website.
So now it's my pleasure to welcome our keynote speaker, which is Chi Anji, and she will be speaking to us today about phenotypes, genotypes, and voxels, a playground next to a nuclear power plant. We're very interested here, and welcome, Chi.
Dear audience, thank you for joining me today. My name is Chu Anqi from National University of Singapore. Today, I will talk about phenotypes, genotypes, and voxels, a playground next to the nuclear power plants. Recent growing population neuroimaging study allow us to investigate massive association between brain imaging measures and health outcomes. And such association can provide insights of brain development and aging in link with physical health, blood assays, lifestyles, such as the one shown in the UK Biobank study. Given the large dataset, there is also a great opportunity for us to investigate genetic markers associated with brain imaging phenotypes, and hence to understand biological mechanisms that are related to brain structure and function. In this talk, I will discuss how to examine association of genetic data and brain phenotypes when the sample size and data dimensionality are large. What are the challenges and possible solutions? In addition, when the sample size is small, but the dimensionality of the genetic data is large, What are the challenges and solutions? I will introduce several solutions to deal with genetic data, such as haplotype analysis, polygenic risk scores, and gene site analysis. Finally, I will touch on gene expression from postmortem brain in relation with brain imaging phenotypes. Genome-wide association is the most popular approach to examine the relationship between genetic variants and brain imaging phenotypes. Here, the brain imaging phenotypes can be scalar measures, such as structure volume, or voxel-wise imaging phenotypes. Currently, the workhorse tools of GWAS on neuroimaging are mass univariate, where a regression model is fit separately at each imaging measure. In this regression, brain imaging data is considered as a dependent variable Y, and genetic variant is considered as an independent variable M. And there's a set of demographic data, such as gender, age, and education, and these variables are considered as confounding variables. And regression model will be able to model the link between the Y brain imaging measure and the genetic variables M. And this analysis has to be repeated for every pair of brain and genetic measures. Here shows the Manhattan plot. And this Manhattan plot shows the significance level of association between the individual SNPs and the putamen volume. There are only few SNPs whose genetic variants are highly associated with putamen volumes. When dealing with voxel-wise imaging measures, we need a massive statistical test. Hence, a statistical approach to deal with correction for large number of statistical testing are needed. In addition to that, big data also have bigger confounding troubles. What is a big confound trouble? In this regression model, we assume we have included a complete set of confounding variables. Hence, this residual estimation is proper, and such as we have a proper statistics for the coefficients that relative to the relationship between the genetic variants and the brain imaging data. However, in reality, and the selection for X is based on our prior knowledge, or sometimes we may not even measure the complete set of confounding variables in our experiments. And in this situation, and the residuals could be inflated, and such as the statistical results for beta down here, it could be deflated as well. So in such case, and the conclusions from this linear regression can make our hypothesis tests conservative. And sometimes the missing covariates Z down here, it can be correlated with the genetic variable we are interested in. And in this situation, our statistical findings could be positive or negative bias for the association between M and Y. The one way to solve this problem is to reformulate our regression model by inserting missing confounding variables Z down here. Where X here is no confounding variables such as gender, age, education, and based on our prior knowledge, M down here is genetic variants we are interested in. And Z here is missing confounding variables we're going to estimate from this regression model. And beta Z is also an unknown coefficient that indicates the relationship between the missing confounding variables with imaging measures we're going to study down here. So there are many ways to solve this problem, where partially the model is known and partially the model is unknown. So we have proposed one of the ways to solve this problem in this paper, and software is actually available in this website. You can download it. Let me give an example to illustrate the importance of estimating missing confounding variables in GWAS study. Here, we are particularly interested in epigenetic markers. We are looking at how our body remembers what happens to us previously, and how does that affect brain development. For each of the units in this study has both diffusion tensor imaging data as well as methylation lossing. We consider fractional anisotropy measures at each voxel as y in this linear regression, and each methylation markers is considered m, and confounding variables we know includes gestational age at MR scanning time, gender, as well as the birth weight of units. And Z down here is missing covariates, missing confounding variables we need to estimate. And this regression model has to run for every voxel in the brain as well as every methylation markers. What we found is that these particular CpG markers that are near to the gene SMOC2 is actually associated with the FA values in the thalamus region. And the thalamus region is particular, has a high concentration of calcium. During the estimation, we found the two missing confounding variables across the entire neonatal brain. And these two confounding variables are actually highly correlated with neonatal birth length as well as total brain volume. So in our study design, we never sought birth length and total brain volume that could influence the association between the epigenetic markers as well as the neonatal diffusion measures. And this is point out the importance of estimating these missing covariates, and such as that will be able to increase the statistical power in the association study between the epigenetic markers as well as the brain imaging data. Beyond the confounding problems, there are other challenges in GWAS study. For instance, common genetic variants only account for a small fraction of the heritability of brain phenotypes. To give an example, this COMT WAL158 match SNP. It's a very popular SNP that people often study. The reason because the COMT regulates dopamine signaling in the prefrontal cortex. And this particular SNP leads to amino acid change from valine to methionine at position 158. The WAL allele is suggested as a predominant factor that determines higher COMT activity in prefrontal cortex, which presumably leads to lower synaptic dopamine levels through enhanced degradation. This particular SNP indeed modulates the association of antenatal maternal anxiety and the sickness of inferior frontal cortex. But there are other SNPs in the COMT genes, and sequence variants lying in the other loci within the COMT genes also affect the gene function, ultimately resulting in altered enzyme activity and adding complexity to the functional as well as the clinical implication of COMT variation. To further examine additional two COMT SNPs, and here we show these two SNPs also modulate association of antenatal maternal anxiety and cortical sickness of superior frontal cortex as well as parietal cortex. Certainly, we can repeat the analysis to every SNPs on the COMT genes. Nevertheless, the genes normally have a nice block structure called the haplotype block. Within each block, the genetic variants of these SNPs are highly correlated with each other. In other words, the genetic variants of one of SNP can represent the genetic variants of this entire block. For COMT genes, there are 3 independent blocks. And using these 3 blocks, we can form a haplotype for this particular COMT genes. And this approach allows us to reduce the number of statistical analysis we need to do in order to examine the entire COMT genes, but in the meantime, it increases statistical power. For instance, using the same data, if we look at the haplotype, and this haplotype actually modulates the association of antenatal maternal anxiety with the cortical thickness of inferior frontal cortex we see before, and superior parietal cortex, as well as superior frontal cortex. So you can see the regions involved is much larger than what we see before when using individual SNPs. But in addition, statistical power in terms of p-values is much stronger. What we learn from the haplotype analysis is that considering the gene information as a whole rather than individual SNPs can improve statistical power. In other words, accumulative effects from individual SNPs can improve the detection on the heritability of brain imaging phenotypes. The second approach we will introduce is called polygenic risk score. We will give an example to illustrate how to compute the polygenic risk score for individual subjects. In particular, in this particular example, we investigate how genes and antenatal maternal depression affects fetal brain development. The dataset includes neonatal brain images as well as genotype data. The risk alleles and effect size of SNPs are established from existing genome-wide association study for depression using relevant discovery samples based on their p-values below a defined threshold. Polygenic risk score is calculated for individuals in our sample as the sum of the count of risk alleles weighted by the effect size in the discovery sample. Previous study showed the polygenic risk score for major depressive disorder predicts the risk for depression and modulates the influences of childhood maltreatment on depressive symptoms. Our study also showed significant interaction between antenatal maternal depressive symptoms and neonatal polygenic risk scores on the right amygdala volume. Neonates with high polygenic risk score and born to mother with higher depressive symptoms show a larger amygdala volume. We employ gene network analysis and identify biological pathways that can potentially contribute to such an interaction. The genes that form glutamate receptor activity most interacts with maternal depressive symptoms on the neonatal right amygdala volume. Postmortem studies with major depressive disorder suggest altered glutamate synaptic signaling is a treatment for major depressive disorder. The antidepressant blocks stress-induced increase in glutamate release in the amygdala and modulates a behavioral response to stress. This example illustrates the use of polygenic risk score in imaging genetic study and its potential statistical power to detect polygenic effects on brain imaging phenotypes. So far, I only introduced a few solutions to the first challenge that is related to a common genetic variants. Beyond the common genetic variants, we know rare variants account for missing, perhaps hidden heritability, which is not even considered in GWAS due to a lack of statistical power. Rare variants can only be identified using GWAS when a sample size is extremely large, which is difficult to achieve in imaging genetics. Is there any possibility to study rare variants effects on the brain, given the current the sample size we have? There are some ways to do it, and for instance, we can consider gene as gene sets. And regardless of what SNPs is inside, whether it's a common variant or the rare variants, we're going to all include into this gene set. We can formulate the effects of genes interaction with environmental measures, for instance, in the following way. And here we have gene size information, and E down here is environmental factors we have down here. And in order to look at the interaction between the two, and we have the interaction terms down here, where E is environmental factor and G is gene size. W here is the weight measures for individual SNPs, and you can think of, and this can be adopted from the GWAS study, or maybe this is just related to the relationship between the SNPs down here. And these two measures actually can generate genetic scores that will be able to interact with environmental factor. And pi down here is the coefficient that related to how strong the interaction between the two and contributes to the brain imaging phenotypes right down here. And we also introduce random effects. For instance, if individual SNPs has a different effects to the outcome measures, and the delta down here will be able to model these heterogeneous effects from individual SNPs in the gene sets. And this approach allows us to be able to combine the rare variants into the gene size, regardless of what the sample size we have. To give an example, and here we are interested in looking at FKBP5, particular genes that regulate HPA axis. We are looking at its modulation to the relationship between maternal depression and neonatal hippocampus. The data sets we have is 161 mother offspring diets, brain imaging data, as well as the genotype data. If you look at individual SNPs in these particular genes, and some of the genes, some of the SNPs actually have a positive association with outcome measures, and some have a negative association with outcome measures. And certainly, this is a heterogeneity of effects from individual SNPs in these particular genes. And this model we just introduced are able to model such heterogeneous effects through this random effect factor. In the meantime, we also be able to look at homogeneous effects from individual SNPs that contributes to the outcome measures. And we look at that here. So what we found is that for the neonates with a higher genetic risk, and if the maternal depression symptoms is high, the hippocampus volume is actually larger. And this is consistent with what we just showed in the previous slides, that the right middle volume is actually associated with larger maternal depressive symptoms. So this actually gives us quite a bit of strong statistical power to look at the gene as a set of genes together in order to boost up the statistical power. You may ask how to choose a gene set if I don't have a specific hypothesis. We all know gene is expressed in specific breed region at specific timing. If we know the breed regions we are interested in at a particular lifetime, we should be able to use the gene expression information to locate which are the genes that are most interesting in this particular region and particular time. In the next few minutes, before I end my talk, I will discuss that the neurotranscription information is actually highly correlates with variation of cortical morphology. Such correlation actually can reflect the biological process related to brain development. Here we extract gene expression information from a brain span database, and we'll be able to actually measure the gene expression information at specific brain region and from fetal stage to early childhood. So we then have brain imaging measures, including structural MRI as well as diffusion imaging data from neonatal and to early childhood. So here we show the cortical thickness was associated with a glial dendrocyte gene expression during trimester 1 and actual site expression during trimester 1 and 2. And fractional anisotropy measures was associated with gene expression in the microglial cells, particularly in trimester 2. So we then take top 10% of genes whose expression are highly correlated with cortical measures. We basically looking at their biological processes that contributes by these genes. And this biological process particularly involving cell migration, cell proliferation, as well as vascular development. And those information are particularly happening during the fetal stage. In addition to that, and here we show that the genes, gene expression information in neurons as well as in astrocytes is highly correlated with cortical thickness data in 4 years, 6 years, and 7 years. And similarly, we see Fe values as well as radio diffusivity measures highly correlated with gene expression information in neuron and astrocytes, particularly in 4 years and 6 years. Again, the top 10% of genes from this high correlation analysis, and that basically contributes to the synapse signaling information in early childhood. And to summarize, and to investigate the relationship between gene expression information as well as the morphology, we are able to identify biological processes that are related to brain development. And this is very similar to what we have discovered in the postmortem analysis. To summarize what we have discussed today, so I basically talk about some of the challenges related to confounding variables. The sample size is small, how we deal with common genetic and rare genetic variants in the imaging genetic study. We introduce a couple of solutions that allow us to estimate the confounding effects. In addition, we also introduce a polygenic risk score, HYPLOTYPE, as well as gene set analysis to study common as well as rare variants together. Finally, we discuss gene expression information is very important for us to look into it. And since the gene expression information encodes both spatial as well as temporal information of the brain, and using this approach that allow us to pinpoint when the functional gene, when gene is functional, in which location in the brain, so that allow us to be able to study gene in relation with the brain phenotypes in a better manner. And finally, I'd like to thank my lab members who have been contributing to the work I discussed today. And thank you for your attention.